Okay, let's get right back at it. This is our 11th mini lecture for the urinary system. So for quote unquote today's set of lectures, I basically want to get through our next objective. So I'm going to do just a few slides in this lecture. The next mini lecture will cover water conservation and how exactly that happens, and then overview or review the entire process of urine formation. And then the last set of lectures for quote unquote tomorrow, whatever that may be for you, is going to be specifically on how we regulate urine production. I think that that's sort of a nice breakup. And then after all of these mini lectures, we will have our third exam. So you should be getting more than enough email contact from me before then to make sure that we all know what's going on. But essentially, it's still going to be the same format as what we had before. I'm trying to keep things as consistent as possible for all of our sanity. It'll be 50 questions. You'll have 50 minutes to do it. It will be on MO2. It'll open during normal class time if for some reason you need an exception to that. So you can't take it from 8 to 8:50. Please let me know as soon as possible so that we can make sure that I have it set up in a way that works for you. If you have other accommodations that I need to know about, again, the sooner you tell me about that, the easier it is for me to make sure that everything goes smoothly. So, yeah, and it is closed note, closed book as before. I am putting a level of trust in you that you will not engage in academic dishonesty, and trust me that we will make sure that everything goes smoothly with that too. So again, you'll be getting some more information from me, but I did want to just throw that out there in case you haven't paid attention to your emails to this point. Okay, let's get going. So at this point, we know that there are a number of solutes that are going to be filtered in the glomerular capsule and then need to be reabsorbed in the peritubular capillaries. And we've sort of just looked at that process of how that happens. One thing that we haven't touched on is this idea of how much of a solute is going to be filtered and then how much of it can actually be reabsorbed. Over the course of this process, we sort of have to have a balance. There are a limited number of transporters for each of our ions or each of our molecules to move them from the filtrate into the tissue fluid and then into the capillary. We're going to use glucose as an example here. In a healthy individual, we should see glucose-free urine, right? Anytime we have glucose present in urine, it indicates that something is wrong. The way that this works is what we have here is sort of a simplified image, right? So this would be our afferent arteriole or glomerulus and our efferent arteriole. Here's our glomerular capsule. We see green dots are glucose. In a healthy individual with a normal blood glucose concentration, some of that glucose is going to slip through our filtration barrier, filtration membrane, and it ends up in our filtrate. So it's in our filtrate, or in that fluid, our tubular fluid. It's going to move through the proximal convoluted tubule, maybe into the distal convoluted tubule, but along the way, it's going to encounter cells that have glucose transport proteins. Those cells are going to respond to the presence of glucose by picking them up and kicking them out. So right here we have a glucose that's binding to a protein, a transport protein, and then getting kicked out where it can be picked up by our peritubular capillaries. By the time the filtrate makes it into the ureter, it should have no glucose, right? So these transport proteins have done a good job of picking up each of these glucose molecules and kicking them out to be picked up by our capillary system. If we have too much glucose, right? So here's somebody who has hyperglycemia or has a very high blood glucose concentration. In the filtrate, we get an incredibly high concentration of glucose. Normally not a huge issue. As it moves into the renal tubule, these transporters are going to pick up as many glucose as possible. But if we look at this image, we notice that every single one of these transport proteins is currently occupied. While they're all occupied, this excessive glucose load, all these extra glucose are going to slip past because they simply cannot bind to a transporter that's already filled. So they make it past, and what ends up happening is in our urine, we end up with glucose. Okay, so this situation where every transporter is blocked or is currently picking up and moving a glucose molecule is called saturation. Okay, so every transporter is saturated with a glucose. While it's handling one glucose, it can't make the other glucose like wait in line. So those glucose just keep moving as they're pushed through the renal tubule and they end up getting excreted. Any excess of the solute is going to now end up in the urine. Okay, so every solute has what's called its own transport max that is entirely dependent on how many transport proteins are present versus how much of that particular solute is present. So as long as we have an appropriate ratio of transporters to solute, we should end up with a fine concentration in the urine. And I think, let's go to the next slide. I think I have a table here in a few minutes that shows us, it sort of reviews what we should see in urine and what we shouldn't see in urine. Okay, that must be on the next slide. So this is a recap of what's going on in each of the locations from proximal convoluted tube through collecting duct. And I realize we haven't gotten quite to this one yet, but I did want to show an overview and sort of walk through what we expect to be happening where. So here's our glomerulus. Right here is our glomerular capsule. So we have filtrate moving into the proximal tubule. The proximal convoluted tubule is going to be doing a lot of our reabsorbing. So what we see here is active transport arrows are shown in red and passive transport arrows are shown in, let's call that lavender. So we get passive transport of bicarbonate ions and water and potassium out. So we get reabsorption. We also get active transport of sodium and chloride and other nutrients. So this would be like our glucose, right? All of those things are going to be kicked out of the proximal convoluted tubule to be picked up by our peritubular capillaries. We also get active pickup or secretion of hydrogen ions and some of our nitrogenous waste products. So they are going to be picked up and put into our filtrate. As we move into the descending limb of our nephron loop, we see that we secrete water. Down deeper, we're going to secrete sodium and chloride, and then up higher again, sodium and chloride, so that's in our thick segment. As we move into the distal convoluted tubule, it looks fairly similar to what we see in the proximal convoluted tubule, although different concentrations at this point. So we're still going to be secreting a bunch of water and salt and bicarbonate, and we're still going to be picking up, or we're in turn going to be picking up potassium and hydrogen ions. So you can see some of these we secrete or we reabsorb at one point and we secrete at another. It all has to do with concentration and chemical composition of the tubular filtrate at each point. As we move into our collecting duct, we're primarily excreting water at this point or doing our water conservation. So in the proximal convoluted tubule, about 65% of our filtrate's reabsorbed here. In the nephron, we reabsorb another 25%. That leaves us with not a ton, but remember, still 36 liters by the time we're in our distal convoluted tubules, mostly water and salt going on here. Again, we have that hormonal control. By the time we're at the end of our distal convoluted tubule, we have urine as far as its chemical composition is concerned. So once we're in the collecting duct, we do very little changing to the chemical composition of the fluid, but rather we are mostly just absorbing water there. The distal convoluted tubule is also going to be involved in removal of drugs and waste from the blood and secreting them back into the tubular fluid. That's not really clearly shown here, but anytime we do have excess waste products or toxins in the blood, we will push them from the peritubular capillaries into that distal convoluted tubule. That's sort of a last-ditch effort to make sure everything gets moved out into the urine. So here is the table that I referenced 2 slides ago, looking at substances. What we have is each of the substances of interest. We have the amount that is filtered, so this is what ends up in our filtrate at the glomerulus or the glomerular capsule. We have the volume that is reabsorbed, so sent from that filtrate back into the blood, and then we have what actually ends up in the urine. So we know with water, we start with 180 liters being filtered. Most of that, so 179 liters, is reabsorbed, and then 1 liter is excreted in urine. Proteins, if we think back to, I think, our very first lecture, I went through what we should and should not find in urine. Proteins should be 0. So anything that gets filtered is going to get reabsorbed. Chlorine, you can see we actually reabsorb most of that because that gets pulled along with sodium. So if we compare chlorine and sodium, we'll see that for both of those, we reabsorb most of them, and we usually excrete very little in our urine. So we filter a bunch, but we pull back most of it back in, and that's because sodium causes that attraction for chlorine or chloride ions, and so we should see similar patterns here. Bicarbonate, we use that as a buffering system. Remember, that's that HCO3 minus. We do filter quite a bit, but we reabsorb most of it, so very little ends up in urine. Glucose, again, we filter a bunch, but really we should see none. As long as you have a healthy blood glucose concentration, that value should be 0. Urea, uric acid, and creatinine, we do filter a bunch. We also should secrete a bunch. Right? And then potassium can be kind of variable depending on the status of the body, but most of that does get reabsorbed, and we just excrete a little bit. Okay, so we are going to come back to water conservation in the next mini lecture, so I will end here.